we'll be having a presentation first by Professor Maxwell Mwebu, and then after that, we'll have a sharing of experiences from some of our sites that are currently undertaking clinical trials. That will be led by the director of CESTA, Professor Obiageli Nodu. But please ensure that you sign the attendance. Let me just repeat that. And I will appreciate admin. Please keep putting the attendance link. The attendance link is on now, please, so try and sign in. And in a minute, we will have Professor Maxwell Mwebu of University of Abuja, who will take us on the training, laboratory procedures, and sample logistics quality management. Over to you, Prof.
Good evening, everyone. Thank you, Mr. Ajayoga. Please, can the admin team give me co-host status so I can share my slides? Okay. Thank you. Let me see. Let me try and share my slides. I hope my slides are visible.
Yes, sir, they are visible, sir.
Okay. Good evening, everyone. This presentation was to have been taken in an earlier week and is a very short presentation before we go into the main thing for the day, which is the discussion that will be led by Professor Nodu. It's going to last basically about 15 minutes in the original schedule. That's what it's supposed to be. This is just to give us some idea of some of the things that we need to be conscious of, especially as we are preparing to become sites for clinical trials. One is that regarding quality management in terms of laboratory processes and also logistics, there are certain components that are very important. One, personnel. Other components are your lab workflows, how you set up the lab workflow. Then we need to be conscious of the specimen life cycle, and this includes things like the courier services, the storage facilities for samples that wouldn't have to be shipped on a daily basis, what we call backup samples, especially when you're dealing with PK studies or PD studies. That is studies that have such components. We need to be conscious of the visit days, things that are needed like the dry ice and the rest of them. Those are all things under the specimen life cycle. Then, of course, normal quality assurance or quality management requirements, which is a key component of any lab at any point in time. And then we also have to be conscious of the regulatory expectations, what the regulatory bodies will want to look out for when they come for audits or site evaluations, whether it's NAFDAC or especially NAFDAC actually, because it's NAFDAC that will look at certain things, especially the lab aspects in terms of tests and evaluations done. So those components under regulatory expectations will be things like our SOPs, our training logs for this personnel that I've already listed at the topmost part of the slide, our calibration certificates in relation to the equipment that we're using for the biosampling preparations and for shipment and all that. So those are the various components that we have to be mindful of when we are thinking about the lab processes for a clinical trial. But at this point, it's important to note that the typical protocols that will be shared by either the sponsor or the sponsor basically, and even the workflows that the CROs will bring from the sponsors or the labs that represent them, we have to know that sometimes they will not give you some practical things that are required. Not because they want to deliberately hide it away from you, but it's just because that's the way protocols are supposed to be. There are some things you need to think out and arrange by yourself. So I'll give an example for what I mean by that. If for example, they tell you, oh, a particular set of specimens must be shipped on a daily basis. That means if the participant comes into the for the visit, you collect that sample, it must leave the country to the central lab that same day. Nobody will tell you how to handle the logistics of how you're going to do that. They will just tell you, oh, this is the courier that we've engaged for this. But every other thing that has to do with it, how you liaise with the courier, how you make your arrangements of ensuring that the courier gets anything that you need on site, be it dry ice or whatever, is not written in the protocol. What it means is that there are so many things that we have to be conscious of and the people that we're dealing with. So when we say, for example, the specimen lifestyle and we talk about the courier, we have to ensure constant communication with the courier in order to ensure that we have efficient lab processes. Okay, so I will start off by giving just what I'll call plausible or helpful advice. One is, if you're starting off as a site for a clinical trial, please try to insist that you want to have a sub-investigator who will be handling the lab aspect. I must say that sometimes some sponsors may not want to pay the stipends that are given for sub-investigators. They will not want to include a sub-investigator for lab in that payment. But whether they are willing to include that as part of the budget or anything, please ensure that you as a person at your site, you have a sub-investigator who should oversee your lab processes. And I will advise that you don't presume, because, for instance, if you have a clinical coordinator and, for instance, that person is a laboratory physician, you will expect that such a person can additionally function in that role. I will tell you that it may be difficult for that person to go, because even as a sub-investigator clinical, that person already has a lot that the person will be handling together with the PI. So it may be difficult, especially for some of the timelines and things that need to be taken care of. Some things may slip under the carpet because that person has some additional roles that are playing. So it's important that you try to dedicate someone, of course, who has to be a lab personnel, to be a sub-investigator for lab, whether it's paid for or not under the budget. Ensure also that you have dedicated lab personnel. You could use your residents if your residents are not, if you have adequate number of residents, because with the way things are now, some centers, you may just come into some units and you see just one resident, and that's the person who is running around doing a lot of clinical. If you have adequate number of residents, you can engage them. Laboratory scientists, technologies. So you have people who are dedicated as personnel to anchor the laboratory aspect. Okay, and those are the ones that usually, when they're asking for the log, the employee kind of log, those are the ones that you will be listed there, because they will have to undergo the various trainings that most people who are on the clinical trial are going to do, the virtual trainings and every other kind of training. Okay. In addition, you must identify other members of the multidisciplinary team, because you have to work with a lot of people. If you're in a center, you have to identify, depending on the protocol, if it's a protocol or a study that requires cardiologists or anesthetists or whatever, please, you have to identify and engage them and let them be part and parcel of your team. Okay? Because, for instance, there will be ECG requirements, echo requirements in some forms of studies and all that. You must ensure that you have someone that you've engaged in that respective field who should help you with. This is very important, especially when there are situations where you are struggling for time, situations where you need people to go outside of the routine service processes in the hospital to help out with particular visits, especially when you're time bound with it. When you have such people, then it helps you because basically your participant is not going to go and hold on and be waiting just like every other patient is waiting. This is important, especially for those of us from the lab aspect. Some studies will say, for instance, before you do your blood sampling, ensure that the participant have done ECG for the visit. And then you can imagine your participant goes to the cardio room and is just staying just like every other person. And maybe by the time that they are going to attend to that person, it will be like past 12 noon and all that. That has already affected your lab processes for that day. And it's very unlikely you'll be able to meet up with shipment processes. Then you must ensure that you have backup plans. And those backup plans will involve key instruments, okay? Things as little as thermometers that are used to monitor temperatures within maybe the room or whatever, or temperatures in this case, we are talking about the lab, the fridges and the freezers where you store your this, you must have backup. So such that when, for any reason, there is some dysfunction or malfunction of any of the equipment, you have something you can use immediately so that you don't have a lag period where there is no form of monitoring. Okay, nobody wants to see that when they come for monitoring visits. And then I must advise that you should also keep a bioengineer as a key ally. You cannot run clinical trials using the equipment that you need without having a bioengineer who would regularly come for calibration, help you check recertifications and all that. So you have to have them available such that if anything goes wrong, you know they're available to come and quickly help to troubleshoot and find out what the problem is. And then the next thing is, if you're involved in clinical trial, though this applies to everybody, but even in the lab, as a lab person, as part of the lab processes, you have to be very conscious of your calendar and your times. In fact, you should keep a calendar that you wake up in the morning, you're looking at it, and you've already marked it what you need to do in terms of the clinical trial. If you're in your office, you should have a calendar that you can look at. If you're using your phone for that, that's fine, because the calendar is important, is a tracker for you. One is you're going to be ordering lab kits because you don't want people coming in for visits, and it's maybe a week before that time you realize you don't have the kits for that particular visit. And then you know that if you order it, it's going to be shipped from abroad. It's going to come like maybe a month or 6 weeks, sometimes 2 months after you've ordered it. So you must keep a track of your supplies and then put a calendar when, at this point, I need to order for this and all that. You're going to require your calendar for tracking participant visits because you need to know when they're supposed to come. Let the clinical coordinator, of course, in liaison with the clinical coordinator, to begin to remind them about the visits.
But small, very small, just introduce that to it. You say, you just introduce that to it.
Admin, please do the
Please,
needful.
can people mute, please? So you also need
just click my daisy. Ojo, I can come and see baby.
You also need your calendar to keep record of your biosampling records because there are times, especially knowing that even though you're shipping most of the samples abroad, the central lab abroad may also have a third-party lab where some tests are done. Like for some of the studies that we are doing, okay, most of the samples are shipped to a particular place, but things like hemoglobin assessment for the hemoglobinopathy aspects, hemoglobin electrophoresis and all that is done in a third-party lab. So the samples get to that central lab and they still send it out to somewhere else. It's only by keeping proper records at site that you'll be able to like come up with explanations or provide evidence when there are discrepancies, especially when you have a disconnect between what was sent to the central lab and eventually what goes out to the third-party labs, and maybe samples are missing or something you see and all that. It's easy to track that way, okay? You know, yes, we are using central labs are being used for this analysis, but most of those central labs are also doing other things. They're running a lot of other clinical trials by various kinds of sponsors in the hundreds, also running other samples. So it's important that you keep your records, because it's only your records that can help you troubleshoot or like track or do an audit when a problem arises. And it's those records that when you keep them using your date and time trackers, that's when you'll be able to like really respond when issues arise with particular dates and times. Then it's also important to know that your calendar will be very, like I said, it has to be a key ally that you need to have, okay? Protocol amendments will come. Once there is a protocol amendment and they notify the site that, oh, maybe this set of samples have to start, we need to start collecting them now, or to stop collecting them now. Once that approval has been given, either at the NREC level or whatever, and the sponsors of the CRO have it, once they notify you, you go to your calendar and you mark it. And if you need to do any alteration on the flow chart that you're using for the study, that's the point to do it, so that even those working will not continue in the previous pattern because they're already used to it, okay? So it's important that we keep proper date and time records. And of course, if you're doing PK and PD studies, you must be conscious you must document times when you collect respective samples. So we can't run away from date and time management. Now, in terms of the laboratory workflow, I will suggest that we try to have a dedicated section or unit, okay, which is separate from the service laboratory. The service laboratory, everything goes on. You know, we can understand what it is like a free marketplace. And you don't want to be running your clinic, doing your biosampling or laboratory processes in that kind of setting. Just the same way people try to have a small place that they use, like the pharmacy for the clinical trial outside of the service pharmacy. The lab should also, you try to just get a dedicated section or unit somewhere, some space where you will be doing your laboratory processes. And then, if possible, try and locate all the necessary equipment that you require for the lab processes for that study and any other study within that area, okay? That localization helps with even when you want to like provide certain things, like you want to provide maybe inverter backups and all that. You know that everything is within one confined space and you don't have to like sort of duplicate activities, okay? And one of the things that we also will notice if we have a dedicated place is that, yes, sometimes we may have a research lab that we want to use, but prior to your clinical studies, clinical trials, that research lab might have been in use for something else, okay? And those activities may still be active. So what you have to do at that point is to sort of streamline the activities or schedule adjustments such that you don't have, we have labs where, for instance, IT people are working in, they come to do the IT and all that. When it comes to clinical trial, you want to streamline things and be sure that you limit certain things, especially for days when those trials are taking place. If need be, you ask certain people not to show up. If the other research activities are still ongoing, you can dedicate other days outside of the clinical trial to do that. This is very important because this will help with misplacement of specimens. For instance, we once had a case where our site was contacted and they were asking us whether we were the ones who sent a particular specimen to the central lab. And we checked our records and we said, no, it didn't come from us. And eventually they were able to trace the thing. And, you know, I remember the monitor asking, so how could that have happened? I said, well, my thinking is that that particular place probably had they have other activities that they are doing. And it's easy in the midst of it. You've collected EDTA blood sample somewhere. And you can just go ahead and package it and ship it when it's not part of the clinical trial samples if you're not very careful, or you separated. So those kind of things can happen if you also already in that section or unit, you already have activities going on there. You must find a way of streamlining those activities or scheduling the adjustment to suit the days that you require the clinical trial process to go on. Now we'll move fast now. Then, of course, part of the workflow, if there's no participant, then there's no laboratory process. So you must ensure participant availability, and not just availability, the timeliness of it. Okay, there are times that maybe one of your participants is a student, he has an exam by 8 a.m. and all that. What it means is that if the window period for that particular visit is already like maxed out, and the person has to come at a particular time, then you must ensure that you do that, taking cognizance of the fact that they have to do exams. That means you may have to come in and start the process very early in the morning. We've had situations where we had to basically go and pick up a participant from their house by 6 a.m. because that participant had an exam, a WAEC exam, okay, in order to quickly complete the process, and then the participant heads to the school for the exams. Okay. And then one of the things that we must be clear about is that we must optimize the pre-analytical phase of the laboratory workflow. We know that basically in the lab, quality management has to do with the pre-analytical phase, the analytical phase, and the post-analytical phase. But in terms of clinical trial, the way we are doing it in this environment, especially with the centralized analysis in central labs, which are oftentimes outside of our countries, the tendency is that most of what applies to us in the lab is the pre-analytical phase, because the analytical phase is done somewhere else in that central lab. So it's the pre-analytical phase, and then secondly, the post-analytical phase, okay? But not so much about the post-analytical phase, because you're now working based on the results from the analytical phase that was done in an external place. So, and we know in lab processes that basically most of the errors that we have are in the pre-analytical phase. Usually in most lab processes, they'll say 2/3 to 75%, 3/4 of lab errors arise from the pre-analytical phase. And this is the phase that particularly involves clinical trials. That means we have to lay particular emphasis on how we handle that process. But it's important to know that in clinical trials, the typical laboratory workflow that we are used to can be different, it can be variable, it can have overlap of some of the components. What do I mean by that? For instance, we say the principal thing here is the pre-analytical phase. But you see, the instruments that you require for the pre-analytical phase, basically things like centrifuges or freezers and the rest of them, are things that will require calibration, which is an analytical phase process. Majorly, the way we think of it in the lab, that, oh, it's the instruments you use for analysis that you're going to ensure their calibration and all that. But you see, in terms of clinical trials, even the ones you use for the biosampling require calibration. Okay, even controls. There are some studies, especially PK and PD studies, where you are required to constitute certain stock solutions that you add to the specimens that you're collecting, okay, before you ship them out. That is a part of what? Controls, which normally are under the analytical phase. So it's important for us to know that there's some form of difference when we are dealing with clinical trials. Now, we come to our sample collection procedures. Just some pertinent points. One is we must ensure correct labeling, subject ID, date, time. We must use protocol-specific collection tools and guidelines. You see, for clinical trials, there's nothing like from my experience or, oh, this is how I wanted to do it because it's convenient. You must follow the protocol. You must follow the protocol, okay? And then you have to adhere to the SOPs and, of course, the good laboratory practice guidelines. But I want us to note that sometimes the SOPs that we have in our routine lab analysis or the algorithms that we use may be a bit different from the clinical trial. That means we must always follow that which the study, the sponsor has provided, and follow it stringently. we must ensure that, look, we can that we don't have to like try to do everything by ourselves. If you're struggling with a collection of biospecimen from in a participant, please, there's nothing wrong in asking for help. The phlebotomy unit in the hospital is very adept in that kind of thing. Okay, we've had occasions where we've had, especially since we are dealing with most of these studies now currently have to deal with sickle cell disease. We know sometimes we can have difficulty with venous access for some of them. Please, no, don't hesitate to use the phlebotomy unit. But as you're going down to the phlebotomy unit, please ensure that they follow the flow chart and they draw it's called the blood draw pattern, or the sequence of specimen tube collection. Okay, that they follow it. It's not that the man feels, oh, EDTA is handy here, he puts it there before he collects the one for coagulation, sodium citrate. You know, that's already a deviation of the blood draw pattern. Okay, so as you're going, you're there telling him this is the sequence, and you arrange it for him the way he has to do it. Please, at this point, I want to also mention that, look, selection of participants, if you're looking out and you decide that you want to look at people who have good venous access, especially when you're dealing with sickle cell disease patients, please, there's nothing wrong in it. In fact, some protocols, some sponsors will include it as part of their protocol to say, look, eligibility criteria, good venous access, because I can tell you it can be very difficult when you have very poor venous access and you're doing a PK study that, look, they've given this person this infusion, you must collect it at 30 minutes, the next one at 1 hour, the next one at 1.5 hours, and you're struggling. So please, there's nothing wrong in saying, oh, please, sorry, we may not use this participant for this study, even though they meet the other eligibility criteria, because this is going to give us trouble in terms of venous access. Okay? And then ensure clarity of the protocol or the collection flow chart that is provided. Please make sure that the laboratory team understands it, and that at every session, every visit, that protocol is spread out. You know, so, some sponsors will bring a flow chart that is physical. Some, it will be electronic. Like in our site, what we did was that electronic one, we project it onto a screen. We have a TV screen and all that. So it's important that you open it up whenever you're doing anything. Otherwise, the tendency is that the lab staff are getting used to, oh, this is normally what we collect and all that. And then they forget. If you have a number of studies and have some similarities, because some of the central labs is the same number, the same kinds of kits that are supplied and all that, there can be mistakes. So no assumption. You open up the flow chart for that visit and you're reading out the sample and make sure that you collect everything the way it is. Now, when I talked about clarity, I want us to just look at this teaser. I want us to look at sex steroids 1 and 2 samples are only to be collected from male patients when patients are on protocol amendment 2. This is actually from a flow chart. I've redacted the other details from where it came from. Now, the question will be, can anyone who is part of this session tonight tell me what he or she understands or interprets as that second instruction? Sex steroids 1 and 2 samples are only to be collected from male patients when patients are on protocol amendment 2 onwards. Please, I would like to quickly get responses, what they understand by that. Is there anyone? I hope I'm not speaking to myself. Hello?
No, we can hear you.
Yeah, so please, is there anyone who wants to make a guess? What do you understand by that statement in relation to males and females?
To wrap my head around.
Okay, so because of time, let's move on, okay? The question I wanted us to see is, where does the female patient step in? How does this statement relate to female participants? If you look at it, there are ways by which that people can interpret this. It could only mean that sex steroids 1 and 2 are only to be collected from male patients, and they can only be collected when that particular site has had approval for protocol amendment 2. Okay, it could also mean that sex steroids 1 and 2 samples should be collected from female patients, whether there's protocol amendment 2 or not, and only taken from male patients when protocol amendment 2 is in effect. So you see that just looking at this, and this is from a real flowchart, that is difficult. And actually, this caused a little bit of problem, but it was easily handled before it got out of hand, especially before we went into critical weeks. Okay, we got to the critical week at the early part of it. So we had to ask for clarification. And they came out to tell us that this actually says, yes, for male patients, please don't collect from them, but collect from female patients. But you and I agree that this could have been framed better to say, look, sex steroids 1 and 2 samples should be collected from what? All female patients. Then you go ahead to say for male patients, please start collecting after protocol amendment 2 has come into effect. But the way it's written, a number of people could interpret it to mean, oh, please don't collect from female patients, but only collect from male patients when protocol amendment 2 has come into effect. So when I talk about clarity of protocol and the flow chart, this is an example, just an example. Now, I'll quickly go very fast now. Some of these we already know. What are the sample handling processes? We require centrifugation. It's important that we follow the protocol in terms of the timeline, the speed, and everything. Let's not just assume, oh, all they're looking for is serum, so let's just spin. And please, if it says 3000 G, use 3000 G. It says do it under refrigerated centrifuge. Please, you do it under refrigerated centrifugation. We must follow the protocol. Even the reporting procedures, okay? If this thing says, please, into this transfer vial, put 0.5 mils. Please put 0.5 mil into that transfer vial, and don't say, oh, we've wasted this person's serum. What are we going to do with the rest? Okay, it's important also for us to keep track of temperature control, shipment. Is it ambient? Is it refrigerated? Is it frozen? And this brings us to sometimes, like, you can have a protocol, and for instance, they say hematology samples should be shipped ambient. And somewhere along the line, because of increased hemolysis, increased UTP, that's unable to perform, they change it and say, okay, send it refrigerated for us to buy additional maybe 48 hours or so and all that. But the initial flow chart and everything still reads the ambient. It's important that when such modifications are done, that's what I'm talking about, the calendar and keeping track, you go and make the changes so that any lab staff will not be looking at the flow chart and still be acting based on previous patterns, okay? I've already talked about blood draw. Now, I mentioned that sometimes they don't tell you everything, you know. One of the major challenges can be logistics, sample transport. One of the things, like I mentioned, is that you must establish good constant communication with the courier. You have to keep track of early notifications. You have to track airway bills. Okay, when they send airway bills for particular studies, please benchmark them, because you may be involved in 1 or 2 other or 3 studies, and it's the same courier, and he'll be sending airway bills. You have to be careful not to use an airway bill for sponsor A for sponsor B, because then it becomes difficult to track that sample once it goes out. Okay, we also have to keep track of public holidays. When I was talking about the calendar, there are so many things. Public holidays, in-country public holidays, and even externally. Public holidays in the sense that you know that during public holidays, customs will not work, so your shipment cannot go. So you have to ahead note it and begin to look at the window period that you have for that participant, how you can adjust, either bringing the person forward or backwards, okay, within the allowable window, and target that visit that way in order to be able to ship. As you're doing that for in-country holidays, you're also thinking of external. The good thing about the external is usually the central lab will send you a mail to say, look, we are going to be on holiday so and so. And if you want them to still receive the sample during a holiday period, then you have to notify them ahead, because they need to now detail a staff to wait for it. Otherwise, you also have to adjust your shipment to tally with their own time. Now, it's important at this point to mention that, look, even when we talk about shipment, most of the time, we have just a window of 3 days in a week, and we must be conscious of it. And those 3 days are Monday to Wednesday. Now, most of the time, most central labs will not want you to ship samples towards the weekend, because weekend staff are not there. Usually there is very little staff to handle it at that. So they will tell you that, look, anything beyond Wednesday, please don't ship any sample. So meaning that if you ship a sample to Europe or somewhere, depending on the airline, on a Thursday, it will get there on Saturday. You've already put that sample in jeopardy in terms of if it's, for example, hematology, that analysis may not occur until Monday, and that increases the risk of hemolysis. So those are all the things that you need to be conscious of, okay? And then, of course, you must keep archive all shipment documents, because that's what you use for audit trails and all that. Now, it's also important to note that even for shipment, your personnel who are involved in shipment must have certification. They must have IITA certification to be able to ship samples abroad. And that means they've undergone the training for various biosample levels, okay? Biosystem 3, 1, and the rest of them. So basically, and just to say that IITA certification can be expensive, but when we had that challenge, you know, some sponsors initially may not tell you that. That's part of what I pointed out. But at some point, they may start asking for it, okay? So when we had that initial challenge at the early times, I remember Professor Nodu, as director of CESTAB, just reminded all of us that there was a training by Mayo Clinic that she had engaged in prior. And we looked at it. It was a free training for shipment and all that. And we went ahead and we did it. And when the when the CRO came for monitoring and requested for that, and we presented, he said, oh, this is fine, because all across many sites, they were also having that problem. They were looking at the cost of being able to do the IITA, which is not captured anywhere. So if you even want the sponsor to pay, assuming they expect that lab staff should already have that certification, they won't be willing to pay for it. So just based on the experience from our site, he was able to reach out to other sites and sell that idea of the Mayo Clinic based one. And that's what those people used to go ahead. Okay, so I will need to be fast now in order to finish. I think the time is gone. Okay, so we need to do proper documentation. Okay, what are some of the things, examples of some requirements by regulators? MOUs. If you're looking at using a local lab, they want to see a memorandum of understanding or contract with that local lab. Okay, if you're using a central lab and for some reason maybe that lab or that organization is bought by another organization, okay, the owners have changed, it's important that you have to ask for evidence from them of transfer, because if they come for inspection and they see your central lab results reading a particular name and then subsequently reading another one, meanwhile when they submitted the protocol to NAFDAC or whoever, it was the initial one that was there, so there must be evidence of transfer. Then we have to keep sample tracking logs. We also have to know that the post-analytical phase has to do with things that have to do with query and flag values. So that means that once we've shipped those samples and we've taken all the pre-analytical procedures to ensure fidelity in the shipment, then we'll wait for the results to come and we have to immediately follow up. Timeliness is also important when there are queries or flagged results, so that we can respond immediately. So what are the common challenges and possible mitigations? Okay, sample mislabeling. This is usually handled if you're using a barcode system. If you use unscheduled kits in the absence of visit-specific kits, then you must ensure that you do electronic requisition. But what it means is that if somebody was supposed to come for week 52, and because you didn't have the kit for week 52, and you used an unscheduled kit, once you record it electronically, it will show as unscheduled visit. Meanwhile, what they will be expecting is week 52 visit for that participant. So what you will do is to ensure that immediately you follow it up with an email asking them from the back end to change it to week 52 and all that. So otherwise, there are going to be a lot of problems. And when they come towards the end for data evaluation or checking the data at the end, that's when they begin to query some of those things, and then it may be difficult to begin to track exactly what happened. Okay, then we require real-time monitoring, especially when we are dealing with frozen shipments and all that, or even storage at site. Now, if there are cases of poor documentation, what do we do? Staff training and enforcement of the SOPs. It is not what you want to do. It is what the SOP says you should do. Okay? And, we get used to certain ways and we feel we already know how to do it as lab personnel. It's not how you feel or how you know to do it. This is how the sponsor wants it done according to protocol. And then we must keep checking our site inventory. That's part of why I talked about the calendar, so that you prevent stock out. So how do you prevent it? Regular checks, starting early requests. And if your stock requests for what we call virtual labels. Virtual labels mean you ask the external lab to send you virtual labels for that particular participant. You give them the details. They send it to you by email. You print it out with the barcode. Then you can even use your hospital-based test tubes to now collect the samples. If it's EDTA, use EDTA as long as you're sure that those tubes are not expired. Then you use the virtual labels they're giving you because you have the barcodes to ship the sample. I think in conclusion, there are so many things, but in conclusion, I just want to say that reliable lab procedures will ensure valid data. We all know that. Strong and efficient logistics will preserve the sample integrity. And then adequate staff and regular training will contribute to quality management generally of the lab processes. And of course, we know that quality management will support regulatory compliance. When they come to site, they see that you have everything properly done. And then lastly, I want to say we should ensure the safety of equipment or instrument supplied by the sponsor or CRO, not just because those instruments are required for the study. For example, if they give you a refrigerated centrifuge, which you never had, you must ensure that people who have no business using it is dedicated to the study. So it should be used for that study. So it's not for someone else working within the lab to, oh, I have so I want to quickly use it. No, you must ensure the safety of those instruments and use them dedicatedly. Okay? And not just because of that, you may at the end, which I'm sad to say, be asked to return the equipment. And I can imagine if by the time the sponsor is asking you to return the equipment that they brought for the study, there are problems with that equipment. It may be something quite expensive. And of course, who is going to bear the brunt of the repairs? You or the site will have to do that. And maybe part of what they may say is, oh, well, the funds that we're going to pay you, we have to deduct whatever for it and all that. So thank you very much. I hope I've been able to give us an idea of what the processes are. Thank you.
Thank you very much, Professor Maxwell. That was very refreshing, and I get the sense that you could speak till tomorrow on this. But thank you. For the sake of time, we will not be able to take questions at this time. I've noted a few things in the chat room. The link to the videos is being posted there. It's at the CESTA website. There will be no slides available, but the videos of each presentation will be there, so you can go back to refresh the training. There's also a note about a survey that's been posted. If you haven't filled that survey, the clinical trial site feasibility checklist, if you haven't filled it for your site, please go ahead and do that. If you have filled it, then don't bother anymore. With that said, I will turn it over to Professor Unodu, Director of CESTA, to lead the next session, which will be in sharing of experiences on clinical trials. And together with her will be, I believe, Professor Brown and Dr. Akinshete. Gaps and challenges of conducting clinical trials in resource-poor regions. Practical experience from ongoing trials at CESTA and other SparkNet sites. So I hand over to you now, Professor Nodu. Thank you.
Thank you Thank you very much, Mr. Tayo. I can see that Professor Brown is online. I'm not sure if Dr. Akishete is online. But good
I
evening,
am now. Good
everyone.
evening now. I'm
Oh,
online.
wonderful, wonderful. So we just want to, so I really, before I start, want to express my sincere appreciation to Professor Maxwell for that outstanding, heartfelt presentation that he has really shared everything that you need to do from the laboratory aspect of clinical trials. And I'm sure that the people who are already conducting clinical trials, we know that what he has said, has a lot of value. So I want to thank him very much. And so at this point, what we wanted to do was to bring the whole training and with the overall goal of helping people to get ready for their clinical trials. I think at this stage, maybe we have Professor Tayo, how many minutes do we have?
Okay, we have about 30 minutes because I would like us to do the post test at about 8:45. Yeah.
Okay, all right. So we just kind of take maybe 5 minutes, 5 to 7 minutes each, just to share our own experience. Professor Maxwell has actually said everything about our center. But what I want to maybe quickly share with us is how we got ready to participate in clinical trial. There are a few salient things. So maybe I'll just use a slide, this slide to kind of show. So 1 second. If I may share my screen, please. Let me see if I'm able to share my screen. Okay, just very quickly, I'm working out of the university. Okay, 1 second, slideshow. Okay, I'm working out of the University of Abuja. That is the contracting organization for our clinical trials. But then the University of Abuja, we are consultants at the University of Abuja Teaching Hospital. So this is the environment under which we are working, a tertiary reference hospital. We take cognizance of the other people, the consultants and doctors and the services that are being provided. And we are also taking cognizance of the other people who are doing international collaborative studies. Actually, we are quite fortunate that we've had Professor Oji, who has been involved in multi-center clinical trials before we started. So we learned a lot from him, and he also helped us. So we have an ethics review board, and it's important to know how frequently your ethics review board meets, so that you will be able to make sure you send in the proposals ahead, applications ahead of their meeting. And so we also, one of the things that we do is that all the staff at the center, they have their ethics GCP and health ethics certifications, and we keep a track and remind people when they are due for the certification. We have dedicated, just like Professor Maxwell said, sample collection areas and secure documents, and we keep, increasing in terms of some of these facilities. We've had a comprehensive standard operating procedure for all aspects of our clinical trials: the IC, the IP, the data management, SAEs, and then we have we do not store our clinic IPs in the central pharmacy. We have dedicated spaces within the clinical trial area, where we store our IPs. And then we also have a dedicated freezers where we store our samples and IPs. We have automated data loggers, and then we also invested in a hybrid solar inverter at 3 points within the hospital where we are doing clinical trial activities. Although we have within the hospital laboratory, which we also take cognizance of, but we are having everything, like Professor Maxwell said, in the research laboratory. We make sure that we maintain annual calibration. It's very painful when you have to pay somebody so much, but we make sure that we do that. And then we also have a refrigerated centrifuge. Now they are collecting it. We have to have always have to have backup. Then we are cognizant of the other facilities that are available within the hospital, such as the clinical trial, other clinical trials that are going on, the facilities that the cardiovascular research team have, so we are cognizant of that. And then for us, we have we are working closely with the research center, and of course, we know we have about 14 clinical trials, that are going on. And then within the hospital, we also know the other clinical trials. But the registry is really very valuable to us. And it is because this registry is existing in other sites that we said, let us have this training so that other sites can also prepare to participate in clinical trial with a goal to making us a destination for clinical trials, in Nigeria for sickle cell. At our center, we have separate specialty clinics for pediatric and adult patients. We have 2 weekly for adults, and then we have, I think, 3 weekly clinics on the pediatric side. We are also aware of other work that is going on, within the hospital, whether it is in HIV and whether it is in respiratory medicine and pulmonology. So we also have a diabetic center, a lot going on in other parts of the hospital, and nephrology. So we have dedicated PIs, coordinators, and clinical staff. And these are important because you can leverage those existing facilities. So this is what we have tried to do. And then, of course, we always keep a profile of our investigators, who can work with us, when it comes to new studies. We have them at hand and we leverage their expertise and experience. So we have a dedicated team of staff, very young people, and also dedicated some senior people. So we have a sub-I for hematology, sub-I for lab, like you can see, and then dedicated clinical coordinators and data management officers. And so this is our experience. So one thing that we have done is that, and Professor Maxwell, Professor Maxwell he's very strict. He manages with the finance officer in the university. He's the one in charge of the clinical trial budget and negotiation and all of that. So when they pay us, we make sure we pay the junior ones, and then we use the money to invest in more equipment and more facilities, because we want to make sure that we have what we need for all the studies that we are doing. So this is our experience, is in occurring in a research intensive setting, and then we are making sure that we have everything we need on the hospital side to help us with clinical trials. Thank you. Thank you. Over.
Yes, thank you, Professor Nodu. And I think if Professor Brown, I'm just trying to see if he's still on. I know Professor Brown was on.
Yeah, I'm still on. Good evening, sir.
Okay, very good.
Good evening, everyone.
You're welcome.
So, thank you for the opportunity to talk. I had asked Professor Maxwell earlier on in the day, and he said we didn't need to make any slides. And Professor Nodo has also talked about the need for us to keep to like 5 to 7 minutes or thereabout. So I'll just go on and share very, very briefly gaps and challenges we have faced in carrying out clinical trials. I have a few slides, but I'll read from my jotter in the interest of time. So, number one is that I think Professor Maxwell did an excellent, probably use a term, I'm forgetting what term she used, an excellent job. Because most of the crises I've been facing, challenges I've been facing, are based on the issues that Professor Maxwell highlighted. Precision in labs things, the cycles, calendar, everything. That is the major thing. And I think if we take attention to all Professor Maxwell said to us, we will go a long way in easing out our problems in clinical trials. Now, number one is facilities. And facilities here are many. One include storage facilities for storing the IPs and also for storing specimen. Now, there's a challenge in that one may not have all the things you need to store your specimen. They may ask for -80 freezers, -40, -20 freezers, and then ordinary refrigerators. So you may not have everything you need. But what I've noticed is that in an academic or research setting like ours, there will be multiple people doing different studies. It may not be clinical trials, but I may have freezers for one thing or the other, including things like refrigerated centrifuges. So as a start, rather than say you don't have, you say you don't have, then you may end up never starting clinical trials. But if you look around your setting, in your institution, I mean, people that may have one or two of those things that you don't have, you may be able to share with, and eventually you have yours. So those are the issues. Equipment for storage, IPs more finicky because they require that they are safe, they are separate, they're not mixed with anybody else. That's restricted access to where they are. So that also may be a challenge. President Odo had mentioned it that you have a clinical trial site. That would be ideal because if we don't have that, we have a site that is not so well, so well ready to take all clinical trials in the entire university and the entire hospital. So it's the hospital pharmacy. So what then happens when there's a strike action or there's a major electrical problem in a hospital pharmacy? Then you may have challenge. So it's one thing I want us to look out for. There's somehow, through collaborations, we've been able to resolve all that. As a clinical trial going on oncology and cancer, they have all the things they need. You talk with them, they may be able to, along with the pharmacists, give you some space where you can keep your own things with also restricted access to your own trial pharmacist without things being mixed up. Because everything must be precise. There are temperature recordings for every investigational product. There's a time and temperature frame within which the drugs or the IPs must be kept. So you must ensure that yours are kept within the time frame and temperature range of the protocol. Again, like Professor Nodu said, you need temperature loggers. Nowadays, there are some digital automated loggers that even at home, you'll be able to know if your drugs in the pharmacy are kept within the stipulated temperature, or if there's a temperature excursion, you have a beep. So right from at home, you'll be able to know that your drugs are safe or your drugs, the temperature has gone out of what is required, and then you know what to do at that point in time. So that talks about temperature recording. They also may want, very often in clinical trials, they want a temperature log. So say in the 24-hour period, what was the temperature like? Perhaps every hour, perhaps every 2 hours and whatever. But temperature logs also, the ones I've had, we have to buy them online. They came from out of Ibadan, some from abroad, but they are very, very useful in ensuring that your drugs or the clinical trial, the IPs are kept safe and are potent. All right, and are potent. The other challenge is power supply. We know what happens in Nigeria, power cord, power supply. So one may need to invest in solar and inverter power systems just to ensure that your samples and also your IP, your drugs are kept safe, regardless of the power outage or power situations where you stay. Like she mentioned a point that when payment is made for clinical trials, which is what I also do, you make sure the junior workers are paid, people are paid. But if one can save some money, some extra money from one trial to invest in this power supply, it will help you and open doors for you to have other clinical trials. In one of the trials we are doing, the sponsors are willing to even procure the facilities of the inverter and everything for us. So sometimes it's good to also ask the sponsor. They may be willing to provide some of these things like power supply, alternative power supply that you may not have in your center. So that's the second issue. The third main challenge is time frame. Clinical trial is everything is time bound. If you see a participant, they may say the record, the data must go into the electronic data capturing within 48 hours or maximum 72 hours, and you must abide by those time frames. And you go outside those time frames, it's a protocol deviation, and it can create a lot of issues. Time frame. Yes, a severe adverse event, so people must be assigned to things. A severe adverse event must be reported within 24 hours of your knowledge. And there are processes, all right? Things to be reported to your local ethics committee, things that you must send to the National Health Ethics Committee, things that you must send even to NAFDAC. And these things are quite a lot. But you see, if people that are being trained are assigned and they know how finicky the CROs, as the clinical research organizations, are, and the sponsors are, then one must be up and doing it, especially in the setting in which people do not take things anyhow. So manpower, there should be no sentiment in choosing people that work with you, because time is of the essence. If you do things 1 day late, 2 days late, it counts against you. It counts against the work, So the same thing with like Professor Markwell said during for PK study, pharmacokinetic studies. It said the first sample was taken plus or minus 15 minutes. Second sample, plus or minus 2 hours. Third sample, plus or minus 4 hours. You must abide by it. And like he said, the lab scientists sometimes, or lab technologies, they just believe they have become used to it. And that's the way to making errors. But the key is to ensure that you never say, I have become used to pharmacokinetic studies, I am used to the timing. Read it out like a Bible. Every time you want to collect the sample, for this sample, patient must not eat before the sample is taken. For these ones, patient must not take the drugs before, or patient must take the drugs, you must take the sample 15 minutes before the drug, or to up to 15 minutes after. One has to read that all the time and place it in the place where the specimens are collected. Some troubles I've run into has been because some people that felt, I become used to it. And in the end, the wrong timing and the wrong things are done. And finally, source documentation. We are used to, working in Africa and Nigeria, especially in Central, where you have so many patients to attend to, and you need to document. And we are very poor in documentation generally in our country, what is so critical. You are doing a clinical trial, you cannot just document the way you document in your routine clinical work. Everything that is done must be properly documented in the patient's case note or case file, as well as the source documents that you are using for the study. So as much as possible, every vital information, because the tendency is you focus on the clinical research source document where you document things, the case record forms, you document things there very well, but your case note does not, mirror or capture the key point there. Again, that causes discrepancies, and that will lead to what they call protocol deviation. It's not easy, I must say, but the best thing there in good clinical practice, if we are used to documenting things properly, documenting time when things are done, the patient is counseled, informed consent, they ask these questions, what are the questions they ask, what are the answer you give. As you document in your informed consent form and documents, in the patient's case note, everything also must be properly documented. They check that there must be consistency between the case record forms, the electronic database, and the patient's case note. It's very, 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 very important. And it's been a challenge. But you see, we must train ourselves to work towards precision. That way, we have less challenges in running clinical trials. I want to say that clinical trials are, I think, the strictest form of research that one can think of, because they're aiming to get reliable data that will impact treatment and treatment outcomes. Thank you very much.
Thank you very much, Professor Brown, for your experience at UCH. Thank you so much. And I will turn it over to Dr. Akinshete also. Take us through a few minutes of your experience. Thank you. I know there is a hand up. When we are done with this, maybe we'll have time for comments.
Okay.
Thank
you so much. Good evening, everyone. I think that Professor Maxwell, Professor Onodo, and Professor Brown, they've practically stripped the whole thing. So I will just want to talk about manpower vis-a-vis capacity development. The first thing is that it is the team that will run the trial. So it's important that there must be good investment in the manpower that runs the team. Every member of the team is important, but some members are more important than the other. So the principal investigator is the person that has the mandate to run the trial. The first thing you need to understand is that when you are doing trials, it is not the same thing as your clinical work. The expectations are different, the language is different, and the output is different. You may be a good clinician in your day-to-day work and turn out and do bad clinical trial. So the first thing that you need to tell yourself is, this is a different thing from all the things I've been doing up until the day you started. You must follow the instruction of the letter. You must follow instructions in the manual to the letter. If they say, do the head before the eyes, don't do the eyes before the head. So getting into trials, you must form the habit of reading the instructions and following the instructions. You will get a schedule of assessment. You get a schedule of how things should be done. So you must follow it. So as a PI, you should try to at least know what is in it. The chances are that as you go along, you can then begin to see similarities in the things that are requested, and you will learn. The second very important person is the study coordinator. I will encourage that if you are forming a clinical study team, try as much as possible not to make a person who has so many responsibilities your study coordinator. Because your study coordinator is the one that is the life of the study. That person is like the gatekeeper, checks what everybody is doing, looks over shoulders, ensures that the process says you must do 1 + 3 before you do 2, and ensures that you follow through on those things. So it is always safer to have a study coordinator that is 100% committed to the trial. It's important. The person must be 100% committed to the trial. You don't want somebody who is going to give 20% or 30%, because the challenge is that when there is an instruction, you as the PI, you also have your clinical duties. And your clinical duties, you want to do them. You want to ensure that you are meeting them. You don't want a study coordinator who would also be as busy, if not more than you. What that would do is that things will fall through the crack. So if you take a physician as a study coordinator, try to have a study coordinator too, who is completely focused on the study. The third person is that you must have a data person who looks at what you or the study coordinator are doing and who ensures that if the study coordinator misses something, then that data person will be sure to pick it up. It is very important because what that does is that the data person is the one who liaises between the study team members, has end to end to look at what was filled in, what is in the source, how did you make a mistake. And when you have people who are not doctors, what it tends to do is that it is annoying. I'm telling you my personal experience because my study coordinator and data person sits there and I'm like, that's not what it says. This is how you do it. They say, thank you, sir. For this study, please write it like this and this. And I find that when I write the way my clinical work is, there will be a query generated. Because the people who are even reviewing what you have written on the other end are largely not physicians. So your ambiguity with words, you're imagining that they are following you. You already have a study coordinator and a data manager who have told you that, look, I don't like your language. Give me a better language. And you find increasingly that you don't have that problem. Now, other members of the team that you need people who are hardworking and who are not motivated by financial gain. It's a thing you need to think about. There are sites in Nigeria who had problem because people imagine that, oh, we're making so much money. How much are you being paid? And that there are some sites that members of the study team went behind their PIs and were speaking to sponsors to ask, how much are you paying us? So you need to also be careful with the people you are recruiting now. They must be hardworking. They must be in need for the work and not the money because they have to first learn how things are done. Prof. Brown mentioned about documentation. It's a problem we all have, whether we're pharmacists or lab people, we must learn to write because in clinical trial parlance, what is not written was never done. So try to get 2 people for each team. So we have 2 pharmacists, 2 people in the lab, 2 nurses, and everything you do must be written down. So a first step for people who are preparing to do clinical trials is to start writing SOPs. Ask Professor Ono this team, ask all the other teams here, can you give us a list of SOPs that you use and SPGs that you use? Because that's the first thing that reassures the sponsor that there is some semblance of science to the things you do because it's written down and it follows a sequence. So what that means is that if you are not there, the next person is going to follow that written sequence and not do it some other way so that there is uniformity and there is a sense of science to it. I think this should just be another. We're already at 15 to 9, and I know the moderator said we should stop by 15 to 9. Thank you.
Thank you so much, Dr. Akinshete. Thank you very much. Interestingly, each of the speakers you have emphasized different aspects that are critical to clinical trials. And we're so grateful to you for that. Yes, the post-test link is being pasted in the chat room and it will be put up again so that while that is up, we'll take 1 or 2 questions. There's a hand that has been up. Admin, can you please unmute Suleiman? El Yazid Suleiman, his hand has been up. While the others,
Hello?
please, yeah. Yes, go ahead, please.
Yes, thank you very much. My questions regarding laboratory machine maintenance. What about laboratory machine maintenance? Actually, I don't know if your presenter talked about it or not because I missed off for 10 or 15 minutes due to network issue here in Sudan. My questions, did you think laboratory maintenance is a significant value during a clinical trial or not?
Okay, thank you. I think it was
Is clear?
covered, but yes, it is clear. I think it was covered,
Okay.
but Professor Maxwell, do you want to answer that
Yes.
quickly?
Yes.
Okay, so thank you very much, Suleiman. So yes, equipment maintenance is very important. In fact, when regulators also come, they also even want to see the SOP for your equipment maintenance, not just the calibration certificates alone. Now, the thing is, depending on the equipment you're using, usually the bioengineers will tell you how regular you should have in between maintenance before the next calibration phase. Most equipment commonly used for the kinds of trials we're doing now requires calibration every year, annual calibration. So if there are equipments that require in between maintenance certification, especially things like centrifuges use a lot of rotors. So they will tell you should have that at 6 months. Okay, so yes, it's important. Thank you.
Thank you.
Okay, thank you. While we're taking one or two other questions, please go ahead and click on the link to the post test. It's in the chat room, and then we'll give room to Ernest Ezeogu. Admin, can you please unmute him?
All right. Thank you so much. It's been a very enlightening session. Thank you for the opportunity to ask my question. So I work in a health management consulting firm, and there's been an increasing demand for clinical trials. However, it is not an area we have strength in. I just wanted to ask if we were to, conduct a clinical trial, what's like the first step for the institution? What do we need to, what are the boxes that we need to check? I mean, how do we even begin? I am an early researcher, so yeah. Please confirm you heard me.
Yes, it's clear. And I'm going to ask Professor Unodu to pick up on this one.
Yeah, thank you very much, Ernest. I don't know if you joined from the beginning, because if you look at what we have tried to do, that is essentially what this entire process has been about. And we have a feasibility, right? If you check what on the chart, we have a form that talks about clinical trial feasibility checklist. So this typically helps you to know what to do, what you need to have in place. And all our discussions, even the presentations today, have been how to get ready, right? The kind of training that you need, and your access to patients. So I don't know if you say you're in a clinical trial, if you say you're in consultancy. I've forgotten how you described your organization, but you need to have access to patients or to investigators who have patients. But they need to have this checklist in place. And these are all the things that they need. So please go to the chart. You will see the feasibility. And then, so there is preparation by the human beings that are going to be involved in the trial. You heard Dr. Kishete talk about the data persons, the clinical coordinators. You've heard Dr. Brown, Professor Brown talk about these investigators, the sub-investigators, the entire spectrum of people, the nurses, the doctors, the lab people. These are all the people that, and more importantly, you have the patients. You have the patients that would qualify for the clinical trial. That was why I showed you, in my hospital, not just us, but other people who are also working within the hospital, because they will form people you can piggyback on, you understand, in terms of personnel, because sometimes the trials are not just in one area, they may involve other areas. So you have to have those people as sub-investigators in clinical trial. It's a whole ecosystem. And for these past 12 weeks, we have tried to lay out what is essential. So I don't know if you were here from the beginning, because these are all the things we have addressed during this 12-week course to enable sites to get ready. Thank you. Over.
Yeah, thank you, Professor Nodu. And I think, Ernest, you can also check out the link to the videos for the 12 weeks, and it will just lay the story also for you, along with the checklist that Professor Nodu has mentioned. Thank you. And so now for the next 5 minutes. All right, thank you. For the next 5 minutes, please be sure to do the post test. And I will appreciate admin to please repost it. Once you click onto it, it's a Google form. So you just go in and then please try and complete that in the next 5 minutes that we have for this session to end. So we just wait on that to happen in the next 5 minutes. Thank you. Okay, I'll give us 1 or 2 more minutes before I call on Professor Onodu to round us up tonight. Yes, I can see some notes about the post test. You know, it's a Google form. Once you open it, it will still be there until you submit or cancel. So once your network is back, you just go back to your form. But please make sure that it's done. If it's not done now, it probably won't be done after this. So please make sure it's done before we round up completely this night. And it's 9 p.m. So in another minute, I will invite the director of SESTA to do the honors tonight as we round up. Someone is still asking for the link to the post test. Okay, for the interest of time, at 9:05, that's in 3 minutes time, I will hand over to Professor Onodu to round up not just tonight's session, but the 12 weeks of intense training. Thank you to everyone who's been part of this the past 12 weeks. So, Prof, please, at 9:05, I think. I will hand off to you. Admin, please mute everyone else. Thank you. Okay, thank you. Professor Nodu, please go ahead.
Yes, thank you very much, Tayo. If you look at what I'm sharing, it's a curriculum that we have used for this 12 weeks, this amazing 12 weeks of learning that we have had starting from the 18th of September. And please, if you can mute everyone, please. 18th of September, we have gone through this entire curriculum that we drew out, and we have had amazing and outstanding presentations from people who are really practicing either as regulators or who are actually doing the clinical trial themselves. So this rich training has been brought to us by IQVIA. They sponsored it in a way that all the people who spoke were given an honorarium, and some we still have to get across to them, especially those ones who are abroad. So we have partnered with BVGH to be able to bring this clinical trial training to us in Nigeria and across Africa. As you can see, that someone from Sudan has been with us from the very beginning. So we have considered all aspects, but what was uppermost in our minds were the areas, the gaps that we had, and the areas that we needed to build a capacity on. So all the videos are the website of Ancestor. So people are free to go back and watch each session, not the presentations, the videos themselves, so that they can watch and learn and also maybe share with others. So I want to express my sincere appreciation to our resource persons, Dr. Akishete, Professor Brown, and all the other resource persons that have presented, during the data persons, Professor Maxwell, Ruben Chanumba, and all the people who presented, because they shared out of the wealth of their experience. You could see the passion with which they shared. And all the sessions have been practical with a view to helping us to actually participate in clinical trials. So we had a site feasibility survey instrument, which we shared on the clinical trial WhatsApp group, but not many people have actually responded to. This is what typically you'll be asked when sponsors come to your site. And it's important to have that site accessibility, the feasibility, because it helps you to know what you have, what you don't have, and what to put in place, even as you train yourself and your staff to prepare for clinical trials. Why are we talking about this? Because clinical trials helps us to up our game as clinicians, the game of caring for our patients. Being able to participate in clinical trials exposes our patients to novel treatments which they would not have. And some of these trials go on for 2 years, 3 years, 4 years, in which the patients, after the initial randomized placebo control stage, go into the open label. And if that drug is working, they will have access to it for quite some time. So we owe it to ourselves and our patients, and our institutions to prepare ourselves to do this. I believe that we can be a destination for most multi-site studies for clinical trials. So please, this is what we went through just to refresh your mind. And all of these talks are at the CESA website. So if you go, you can listen, and then you can gather your people and do maybe like another 12-week training, in-house, and you have access to all the videos. I want to thank Mr. Jayoba, who is engaging with this sponsor to get all of this done. And I want to thank everyone who has been here with us for these 12 weeks. I see the same people every week, and they've engaged with this process. It's been an exciting, I have learned a lot. In fact, if you do this again, people will learn different things and new things, even as we have participated. So I want to thank everyone for being here, for turning up each week. And then we want to write a paper. And the paper that we want to write, if you can see the richness of what has been shared this evening, you can see that if Professor Maxwell puts those things he has said in writing, many people will be able to just access it and use it. If Professor Akishete puts into writing what he has been saying, is the same thing Professor Brown, all of us. So that's what we want to do. We want to use that site feasibility survey to get information about site readiness, and then we will discuss all of these things in a paper that will make us visible to external funder. Yes, we're already participating in clinical trials, but there are other sites who are not, and they may be ready to participate. So we need to increase our visibility as sites to participate. So please, if you have not filled that form, please fill it. It will help you to know what you need to have. It will help you to showcase what you already have, and then it will put you out there for sponsors to contact you and to be able to come to this country for clinical trial. I know that we've had our colleagues from Ghana and from across Africa. I want to thank everyone, and I'm very excited. And I know that this is not the end. I see a situation where other sponsors, apart from IQVIA, we want to make this like an annual thing. And then we are all getting ready, and we are all practicing, and we are all giving our patients the very best. So thank you very much. Thanks to the team at the IT team that have supported this training through the 12 weeks, and the data officers. I really want to express collectively for all of us just to say thank you. Thank you very much. Over.
Thank you,
Yes,
ma.
thank you, Ma. And Professor Maxwell, I think I'll hand over to you for the certification talks, and then we'll close out tonight.
Thank you, everyone. So we are going to use the next couple of days to assess the attendance register so that we check those who made the required attendance. And subsequently, all those who made it, you're going to get emails from us specifying the next steps that we're going to take requiring towards the certificate that will be issued. So basically what we are thinking of is once we have this attendance, we are going to now send to you subsequently via your email, the registered email, the links for the assessment test. So what we've had now is the pre and the post test, basically. But there is an assessment test that we're all going to take that will lead to the certification. So, and you're going to get that notice and then the links via email after we've gone through the attendance registry. So I think that's just the announcement I need to make. So please watch out for the next couple of days. And after the next couple of days, check your emails. You're going to get email notifications from us for those who made the attendance. Thank you very much.
Okay, thank you very much. And then thank you to Professor Unodu. You couldn't thank yourself, but thank you very much for facilitating this for everybody. And with that, I believe we will call it a day in the 12 weeks. Suleiman, do you want to send your comment into the chat room?
Allow him, he's a friend from Sudan.
Okay. Okay.
Thank you. Thank you, Prof. I'm very happy to meet you. Thank you. But I have questions regarding the assessment. What about the score for releasing certificate?
Okay, we
so
will talk about that. Yeah, okay. Go on,
Prof, please
Professor
go ahead,
Maxwell.
ma.
You know, we said it's 100% assessment, and you have to pass. You have to pass the test. Before this time, I had one of the trainings by one of the sponsors, and I answered, I think I got 90, and they said I didn't pass. So I had to retake the test again. So this clinical trial is very important. You have to pass the test. So you're saying the score. The first score is 100% attendance. And this 100% is not if you come in 5 minutes to the time, you say, give me the attendance, because Zoom tells how long you've been in. If you've logged in multiple times, you can get that. So we're going to look at all of that. So it's 100% attendance and you have to pass the test.
Okay. I'm ready. Thank you, Prof.
All right. And I think we can call it a night now. Please ensure you complete your post-test. Just open it up and make sure you complete it. Thank you, and have a good evening, everyone. Thank you.
Good night.
Goodbye.